Um, the we are going to basically discuss the lab today, and the most important thing that you need to know today is the Staphylococcus species that you are going to identify and send me information about it. And that lab is important. It worth plenty of points, so I don't need from you to miss that lab. So this is why I'm trying very much to focus on this lab with you today. So before I start, do you have any question for me, a specific question for me to start with, Raho?
Yes, I have questions. So there was extra credit that we had, and like the last Zoom video that we did, like you said, don't worry about it. But then like I noticed that it went away after June 24.
Yeah. The reason that was
Extra credit.
I was planning for to put this, but I realized there is some extra credit that already placed like maybe a week ago. That would be too much extra credit at this time. This is going to be basically violating, because we have to have a specific percentage for extra credit. So this is why I removed that one. But doesn't mean that we are not going to have any. We are going to have definitely more. But next week, for example, I'm going to put one. But that one, that one was removed. Yes.
Okay, thank you.
Yeah, that is the thing. So do you have any question for me for the lab at this point? Because we said that we are going to specify Fridays meeting for the lab, and Mondays for the lecture, Mondays or Tuesday for the lecture. Okay. So now for the lab, any previous lab that you have any question for me, or otherwise I'm going to jump to the last one, the Staphylococcus identification lab.
No, everything is very straightforward. Thank you.
Great. So let me do this. Let me go and share my screen with you about the staff lab. Okay. So this is the screen. Can you see it? What I need to emphasize and focus on this, basically, what is the main purpose of the lab? The main purpose is being able to identify species of Staphylococcus. You know that there are many species of Staphylococcus. Some of those are pathogenic, right? And some are basically like non-pathogen at all. They are, some of them live in the soil even, for like saprophytes, and feed on dead materials. The idea is to have a Staphylococcus in your hand so you know the genus, but I need for me to identify the species. So you need to go through different kind of biochemical testing to be able to identify the organism. So all Staphylococcus, generally speaking, the genus Staphylococcus, they are all gram-positive, as you know. So gram-positive meaning that if when you perform gram staining, they are going to take the crystal violet color. So they're going to show like purple color. And they are spherical. This is why you call it coccus or cocci, spherical shape. And they are not single, but they are mainly arranged in shapes, mainly in the form of like a grape-like formatting clusters of cells. There are many species. I give 7 species for you guys. Each student is going to perform or to be able to identify that isolate that I gave you. Okay, so all of them share some property, like they are all gram-positive, they are all cocci, they are all cluster shaped. But as soon as you're going to do some biochemical test, then you will be able to differentiate and identify the strain that you have in hand. First test is going to be catalase activity. And catalase is basically, it can be performed as you can see in this picture here. So you have like, you take like a colony, like some scrap, some culture, like from a plate, and then you flood it with hydrogen peroxide substrate. If you see this kind of like effervescent here and the generation of oxygen, that means it is a catalase positive. If it is like that, it is a catalase negative. So this is the first step that you need to look for when you identify. So by looking at the slide, you will see if your isolate or sample is positive or negative based on the figure here. A second test will be for oxidase testing. And again, if it is oxidase positive, the color of the reagent is going to change to purple color or bluish color. If it is oxidase negative, it is going to be negative color. And it is very important to know that those kind of enzymes, oxidase, are basically very associated with obligate aerobic. Obligate aerobes. Obligate aerobes, they have those kind of oxidases. Facultative anaerobic, meaning like staph, for example, some of them, they don't have these enzymes. They don't need it, okay? So if you look at the figure here, when it says that this is your isolate, and you compare between these results here, you can see sure that it is oxidase negative, okay? So now you know it is catalase positive, oxidase negative. You need to put those in hand. Very important media that we are going to use for identification, we call it MSA media, and this stands for mannitol salt agar. Mannitol salt agar. It is a very selective media. Selective because it selects only for those kind of bacteria that can tolerate salt, halogurins, okay? And also it can be differentiated, so can be both selective, meaning it selects the growth of some organism, not the other, and also it can be used for differentiation between organisms. So for example, if you look at the plate and you see growth, so it means that this kind of bacteria can tolerate high salt. And number two, you need to look at the color change of the media. That color change meaning that the bacteria has the capability to ferment the sugar. And the sugar in this media is mannitol. When they do this kind of reaction, they change the color of the media. So based on these results, you can tell that your organism is basically can grow in mannitol. So it can tolerate salt and it can ferment mannitol, okay? So this is the third test. So you need to look at those test by test and to accumulate some kind of biochemical data that will help you for identification. The most important test here when you talk about blood hemolysis. Hemolysis meaning the capability of the organism to lyse red blood cells and form clearing zone, okay? The media we're using for this test, we call it CNA, and you need to know what is the name stands for, okay? You need to know either it is selective or differential, and you need to know if there is any indicator in the media, what is the name of the indicator, and what is the purpose of this media, okay? CNA is Colombian nalidixic acid media, and this kind of media, it is basically, as you can see, red. So it has blood, sheep blood cells, and it has an nalidixic acid. It's an antibiotic which inhibits gram-negative bacteria. So if you have a sample, like say from your skin, that you need to isolate staph from, and you grow in this media, this is selective. So gram-positive only will grow. Gram-negative cannot. Why? Due to the presence of nalidixic acid, antibiotic, okay? What else in this media? There is an antibiotic, we call it novabiocin, and this novabiocin is very, very important because Staphylococcus species, different species can be differentiated about, based on their ability to resist novabiocin. Novabiocin, you have a disc saturated with the antibiotic, and you put it in the agar plate, and you look for what we call inhibition zone. Inhibition zone, because as soon as you put the saturated disc with the antibiotic is going to diffuse in the media. Okay, so basically you take the media, you grow, you straight, like have a, like your organism, you make a very nice streaking to cover the plate very nicely, don't leave any gap, and then you put your saturated disc with novabiosin, and then incubate and look for inhibition zone. Inhibition zone area around the disc where there is no growth. It is critical because you need, as soon as you do this, when you measure the diameter of the inhibition zone, it will give you some information about the bacteria that you are dealing with. We call a bacteria sensitive to novabiosin if the inhibition zone is 17 millimeter or more. So if you measure the inhibition zone and it is 17 millimeter or more, that tells you that your organism is basically sensitive to the antibiotic, novabiosin. If it is less than 17 nanometer, it means it is resistant. So this is a very, very important way to differentiate between different Staphylococcus species. If you grow them in plate, agar plate, like a blood agar plate, there are three major hemolytic patterns. Okay, we call those alpha, beta, and gamma, where beta is making a complete clearance around the colony. So if you have a growth and with complete clearance, as you can see here, lysis, complete lysis, we call those beta hemolytic bacteria. Alpha, they can do some hemolysis, but not as complete as beta. So partial kind of hemolysis with greenish color like this. Gamma, on the other hand, those are non-hemolytic. So it is crucial when you see in your practical a plate and some bacteria, and it will ask you, is it alpha, beta, or gamma hemolysis? You need to look at this zone around the colony. Complete hemolysis, beta; partial hemolysis, greenish color, is alpha; no hemolysis at all, this is gamma. Okay? So looking at your result, you will see that significantly you have a bacteria which is beta hemolytic because you can see nice hemolysis, and you measure the novabiosin, the diameter. Here it tells you it is 25 millimeters. So based on this result, you can tell that my organism is sensitive to novabiosin. It is not resistant. Okay, so any questions so far?
No, everything's clear.
Great. Perfect. Any question? Okay, no problem. So let me continue here. This here very important when you perform some biochemical testing, like to see if your organism is capable of fermenting different kind of sugar. And what I need from you to know from this, let me stop and share another screen. Not that one, sorry. When you have, this is this kind of shape here, you need to know that when we do like a sugar fermentation, we have the media and we have indicator in the media, which give it this kind of color. We have an inverted tube. We call this tube is Durham tube. You need to know the name here because maybe it will give you a picture like this in your practical, and it will point out to this tube and it will ask you the name of this tube. So you need to know that this is Durham tube. And the main function of this tube is basically to accumulate any gas that is, if any, produced during fermentation. So during fermentation, if the bacteria is capable of producing gas, that gas is going to escape and part of it is going to be trapped in the Durham tube. as you can see here. So here you have several different kind of scenario for your result. If there is no change in color, it means that the fermentation is negative for that particular sugar. Bacteria cannot use the sugar. If it is changing color to yellow, meaning that the bacteria can ferment the sugar. If it can change color and give gas, so meaning that the bacteria ferment the sugar and produce also gas that can see in the drum tube. So again, this is very, very important slide that you need to know. Okay, stop this one here and go back to this. Okay, what else I need to tell you here is there are other media in this particular practice which I need also from you to know, and we call this bacteria, this media SM110, and S stands for Staphylococcus, M medium, so Staphylococcus medium number 110, agar. So this particular media is so crucial. Why it is important? Because it has some ingredients in the media that allow your bacteria to make pigments. Some of Staphylococcus are pigmented, and this kind of feature can be used to differentiate between different species of Staphylococcus. So this is why SM110 media is so important in this, okay? The other thing is also in this media, it has also gelatin. And the ability of the bacteria to hydrolyze gelatin, as you know, it's a protein. Some bacteria, they produce some enzymes that can degrade gelatin. So in the SM110 can tell you also if your organism can degrade a gelatin or no. Basically by flooding, after you allow the bacteria to grow and you look for the pigmentation. If it gives you any pigments, usually or always Staphylococcus aureus is kind of golden. So when you see a shape like that, you can tell there is a 95% probability that I'm dealing with Staphylococcus aureus based on this kind of pigmentation. And you can do gelatin hydrolysis testing by flooding the plate with the ammonium sulfate. And if there is a gelatin hydrolysis, you will see very nice clear zone around the colony. Okay, so this is also a very important media. Some Staphylococcus, they have the capability to hydrolyze and degrade DNA. So they produce DNases to do this kind of job. So again, there is a media that is in your test that basically it has the bacteria, and you need to know why the color is green here. What kind of indicator is being used in this kind of testing? You need to know that here the media, it is normal nutrient media, okay, but it has a DNA. This is what you are trying to test for, and the DNA here is basically is coated or attached to the dye, which you call it methyl green, and this is why the color methyl green. And if your organism has the ability to degrade DNA, then you will see nice clear zone again around the growth. Okay, why I emphasize on this? Because pathogenic bacteria or pathogenic Staphylococcus, one of the virulence factor they have DNAs, because as soon as they have the capability to enter the host cell, they can degrade, and this is how dangerous they are. They have the ability to degrade the DNA of the host. This is why some Staphylococcus are so dangerous due to this fact, due to the presence of DNases coding gene in this kind of organism. Okay. Another virulence factor is basically the coagulase. Coagulase is another enzyme produced by pathogenic Staphylococcus, and what it does is to convert the test here is basically you take like a plasma from rabbit, and that plasma is rich in fibrinogen, okay? And if the bacteria has the enzyme coagulase, what they do, they convert the fibrinogen to fibrin, which can cause this kind of coagulation. So you will end up with the plasma, the rabbit plasma, which is kind of liquid at normal room temperature, is going to coagulate and be like a jello like that. Okay, so some dangerous pathogenic bacteria like Staphylococcus aureus, they have this capability. They produce coagulases, so they can convert fibrinogen in the plasma to fibrin that will coat them. So this is what they are doing to protect themselves from the immune response of the host. They basically trap themselves in fibrin. Okay, so the host immune system is not going to recognize them. And then as soon as the condition is favorable, they produce other enzymes to degrade the fibrin and then released again in the plasma of the host, in the blood of the host. This is how dangerous this kind of bacteria are. So both DNase and coagulase, those are pathogenic factor or virulence factor for some staph. Not every staphylococcus has this kind of virulence factor. So again, this is another way of differentiating between the different staph. The table at the end of the presentation is going to be very, very important for you because it will give you some idea about what kind of organism you are dealing with. Again, you look at the data that were provided, and based on the table, you will be able to tell the species of staph that you are working with. Staphylococcus aureus, for example, they have very specific features. One of them is the ability to coagulate. See, it's coagulase positive. They can convert fibrinogen to fibrin. Also, they are beta hemolytic. They make very nice clear area around the cell. They also produce DNases to degrade DNA. They also, when they grow them in the SM110 medium, they convert to golden color, kind of golden pigments. And this is the name aureus, meaning golden shape bacteria. So this is what I need from you to do when you look at this presentation. So you go through each test and you accumulate your biochemical data, and then look at the table that will help you to identify your isolate at the species level. And then as soon as you identify it, then you need to write for me like a 2-page about the feature of this particular isolate that you are working with. So any questions so far?
No, I think that sounds good.
Okay, great. Anything in previous labs?
Not right now. Not for me.
Okay, anybody else? Yeah, as I mentioned earlier, I'd like to do this kind of meeting, and it will be more fruitful, more efficient if you bring your question, or if you email me. Even if you can't attend, it's okay because I'm recording it anyway, and I'm posting the link. So if you miss it, don't worry, you're going to see it. You know, I will send you a link. But again, to make it more efficient, I'd like if you email me a specific question, you know, about how to do this kind of experiment. I didn't get this one, you know, it makes things easy. One last thing I need to go through in the lab is basically, let me stop sharing here and go to, let me do this. And, because this is very important about calculating the number of colony forming unit of bacteria, okay? About dilution and stuff like that. Okay. For bacteria number bacteria count. Not that one. Okay, let me share this with you here. Okay, what I need from you to know from this lab, when you do calculation and like to get the colony forming unit, okay, so what I need from you to know, say I have my original culture here and I need to do standard plate method to count how many colony forming unit per mil in this original culture. So you go through a serial dilution, as you can tell. And in this case here, I'm making like having bottles, each one has 99 ml of a buffer, saline solution, broth media, whatever, okay? And then when I'm moving, say, 1 ml from the original culture to the first bottle here, 99 ml. So the dilution factor in this bottle will be 1 to 100. How did I get this number? To get this number, basically, you have the amount transferred, which is 1 ml, divided by the total volume. Total volume is going to be 99 ml plus the 1 ml that you moved. So that will give you the dilution factor for that bottle. If you move 1 ml from this one to the second one, again, you have a dilution factor of 10 to the minus 2. But the total dilution in this bottle will be 10 to the minus 4. Why? Because you have dilution 10 to the minus 2 times 10 to the minus 2 here. That will give you the total dilution of 10 to the negative 4. Okay? This is what I need from you to know. And then when you do plating, usually you take aliquots. Okay, so you can take 1 ml in one plate and 0.1 ml in another plate. This is very, very important because when you do counting, you need to look at those numbers. How? Say I did my experiments and this is some data here. Okay, so I have before incubation, nothing, and then I have 3 plates, and this is plate number 1, plate number 2, number 3. And this is the number of colonies in each plate. It gives you also amount plated, how many, how much volume did I take from the dilution that I used, and what was the dilution. It is so important to know those information. The number of colony, amount plated, and dilution, total dilution, to do this equation. What plate you need to select? You need always to select a plate which has from 50 colonies all the way to 300. This is the best plate you need to get to count. Don't use plate that has less than 30 colony or a plate that has more than 300. That will be, the error rate will be too much. So always select a plate for this range. As soon as you select the plate, in this case, I gave you the number of colonies, so 280. This is a plate that I'm going to select to calculate my colony forming unit number. Use the equation. Colony forming unit per mil is going to be number of colony, which is 280, divided by total dilution, and this is the dilution I used, 10 to the negative 6, and times the amount plated. And what I did, I used 1 mil from the dilution to plate, so it will be times 1. And then you do very simple math, you will be able to get the colony forming unit. Because this is crucial and important, because you are going to have in your practical, I will give you a plate with some number and I will ask you to tell me the colony forming unit per mil. So you need to have the information. You need to have the number of colonies, you need to have the dilution from where the plate came from, and you need to know the amount plated. Okay, do you have any question for this?
That makes sense.
Okay, great, thank you very much. Anybody else?
I have quick questions. So for the PowerPoint,
Yes, ma'am, go ahead, please.
For the PowerPoint, like, so like for us to do the report, we have to answer the questions like that's kind of leading like identifying the like specific
Yeah, for the PowerPoint, I need from you, this question you answer for yourself, and then because you are going to use this information, remember there is an ID sheet that I emailed. So you
right.
need to take that ID sheet and fill it. It tells you all the biochemical test based on the PowerPoint. And then as soon as you know it, you will be able to identify the species that you are dealing with. Then you need to write for me like a 2-page essay
About that species,
about
right?
that particular species. Yeah, what is the feature, where to find it, is it pathogenic, if it's not pathogenic, if it is pathogenic, what kind of virulence factors are included, how can be treated, what kind of disease can cause, this kind of good information.
Okay, got it. Thank you
Yeah,
so much.
no problem. Okay, so I believe we'll meet again Monday or Tuesday for to go through the lectures. But I prefer to have some questions, so we can go through those in a more efficient way.
Okay, great. And are we gonna have another one about lab before July 4th or no?
Another what?
Oh, another one of these sessions, or is this just like a specific one to the project?
Yes, next week. Definitely we have
Okay.
next week. Yes, we have next week.
Okay.
And I also prefer before the practical to do another review session, for the lab.
Okay.
I prefer to do that.
Okay.
That's
Great.
good.
Yeah, thank you very much. You are doing very good job as so far, and I need for me to continue this good, attitude. You will be fine.
Okay. Thank you so much.
Thank you very, very much.
Thank
You
you
have
so
a great rest of the day.
You too.
much.
Okay, bye, bye, bye.
Bye.